high-yielding bioprocess for Pullulan using novel indigenous strain, a technology developed by Dr. Anirban Roychaudhary from CSIR-IMTECH. Pullulan is a water-soluble exopolysaccharide produced by Aerobasidium pullulans. It is a homopolymer of maltotriose joined by glycosidic linkages. Pullulan is a tasteless, odorless, white powder that is non-toxic, non-mutagenic, non-carcinogenic, edible and biodegradable. The US FDA GRAS status of Pullulan ensures safety and usability across diverse consumer groups. Pullulan is known to be non-comedogenic, meaning it does not block skin pores. Physiochemical properties of Pullulan include distinct film and fiber forming capacity. Films formed by Pullulan are impermeable to oxygen, and the Pullulan molecule can be derivatized easily to alter its solubility as well as reactiveness. Pullulan is an extremely versatile ingredient with capability of providing a technology platform for product innovation. Pullulan has many unique properties, finding application in various novel products, like the film forming and adhesive property helps in making peel-off facial masks with skin tightening and anti-aging effects. Pullulan is also used in making intriguing formulations like the breath freshening strips. Biodegradable thin films formed by Pullulan are used in food packaging to prevent fungal growth by limiting oxygen permeability while retaining moisture content, thereby increasing shelf life of products. The water binding and texture enhancing property of Pullulan makes it a useful thickener and viscosity regulator for various formulations in skincare, personal care, cosmetics, and food processing. The viscosity of Pullulan solution is stable to heating, pH changes, as well as changes in salt concentrations, making it a good alternative to xanthan gum. Pullulan has high stability, chemical inertness, and forms excellent oxygen barrier, which makes it a better alternative to gelatin as well as plant-based capsule shell materials. Market opportunity for Pullulan. Post-COVID global market for Pullulan was estimated at USD 68 million in 2022 and expected to grow at cumulative annual growth rate of 4.5%, reaching USD 89 million by 2028. Key market players include Nagase, Jinmei, Freda, Henbo, Hairan Biotech, and Kang Naxin. The market prices vary from Rs 2,500 to Rs 9,000 per kilogram, mostly based on the quality of Pullulan, which is defined by the color, viscosity, molecular weight, as well as the end use. The advantages of current Pullulan technology for the existing market players are high yield of over 70 grams per liter, which is higher than any reported process. The final product has higher average molecular weight of 280 to 300 kilodalton, which is desired to achieve the unique properties of Pullulan. No melanin byproduct produced in the bioprocess due to use of unique microbial strain. This reduces downstream steps, removing the need for melanin removal, which is present in other bioprocesses used for Pullulan synthesis. This process uses low-cost starting materials, which further reduces the cost of the product, giving a competitive edge. The process media used is optimized to reduce inhibitory effects of high sugar concentration, and the final product is of high quality and comparable to USP specifications. Who should be interested in this technology? Manufacturers of skincare, personal care, and cosmetics, where there is scope for new value proposition for customers. Existing manufacturers of Pullulan can utilize the advantage of the novel strain and process used, giving a competitive edge. Manufacturers of biosynthesized value-added chemicals can have new products and forays into newer segments, giving an opportunity to disrupt the market. Lastly, utilizing the unique properties of Pullulan, manufacturers of packaging industries, food processing, formulation and capsule shells can benefit with new value proposition for customers with a competitive edge. About the technology, the process features a unique strain of Aerobasidium pullulans, which is novel, osmotolerant and non-pigmented strain for production of Pullulan. The process uses low-cost starting material, which is less than $1.5, utilizing agri-industrial residues or a defined process media. The process media has been optimized to reduce inhibitory effects of high sugar concentration on synthesis of Pullulan. The process includes inoculation followed by cultivation in the bioreactor. Following fermentation, the broth is separated, from which crude Pullulan is obtained by solvent precipitation method and further purified to get the final product at a high yield of more than 70 grams per liter and high quality pharma grade Pullulan, which is comparable to USP specifications. The estimated cost is Rs 1000 to 1500 per kilogram. The Pullulan obtained by this technology has been analyzed and found to be in par with the USP specification, like appearance of white to off-white powder and loss of viscosity on reaction with pullulanase enzyme. The kinematic viscosity and pH of 10% Pullulan solution also complies with the USP. Heavy metals, residual solvents or E. coli salmonella are absent in the final product. Current status: the technology has been scaled up to 500 liters fermentation with downstream processing to be fine-tuned. The IP status of the technology is a know-how. About the team and organization, CSIR-IMTECH National Laboratory is the forerunner in the domain of microbial biotechnology research and development. The lead scientist, Dr. Anirban, is a senior principal scientist at CSIR-IMTECH Chandigarh. His expertise includes bioprocess development and scale-up, fermentative production of polysaccharides, and polysaccharide-based materials. Dr. Anirban has over 2 decades of experience in the industry and academia. He has successfully delivered many industry-sponsored projects. Key assets of his team include over 35 publications in bioprocess development and microbial technology, and a team strength of 5 people. CSIR-IMTECH has well-equipped labs and facilities, including lab-to-pilot fermenters of capacities varying from 2 to 1500 liters for continuous and batch fermentation, high-capacity homogenizers, and various other large-scale downstream processing equipments. Few of the many industries that Dr. Anirban has collaborated with are listed here. Next steps. The team has developed the background science and demonstrated technology at 500-liter fermenter scale as early stage validation. The team has expertise as to how the process can be modified to get desired output. Next phase will be to work closely with industry partners to fine-tune downstream processing and carry out late-stage pilot demonstration of the entire process as per industry requirements. The scientist is seeking industrial partners for technology licensing or for sponsoring further technology advancement and scale-up, or even explore opportunity for raising third-party funds for a collaborative project. Interested industries can also reach out for tapping scientist capabilities as an expert or consultant.